Good morning, students. We have already done the nutrition topic, and today we are taking up the three activity concerned with this topic. Now, first of all, you have learned the topic photosynthesis, that it requires the raw material carbon dioxide, water along with energy and chlorophyll, and in this process oxygen is liberated. So the first two activity are concerned with the photosynthesis. First one is given on page number 96. It says take a potted plant with variegated leaf. Now this variegated leaf means a leaf which has two type of patches. One is the green one which contains chlorophyll, and the other is the non green one which does not contain chlorophyll. Okay, now keep it in dark room for 3 days. Now this plant is kept in the dark room for 72 hours. Why it is kept? Because by this process the plant becomes de-starched, that is the removal of the starch, all the starch from the plant, and so the plant is kept in dark. Okay, as it is kept in dark, there is no photosynthesis occurring, and the plant utilizes its stored starch. Moreover, removal of starch is necessary because whatever the starch is left, it may interfere with the result after doing the experiment. That's why plants are kept in dark. Now keep the plant in sunlight for about 6 hours. Pluck a leaf, mark the green areas in it and trace them on a sheet of paper. Now draw the outline of this green and non-green area, okay, over a paper. Now dip the leaf in boiling water for few minutes. Now here you can see leaf in boiling water. Why it is kept in boiling water? Because this process can soften the leaf and break down the cuticle and kill the leaf. It disrupts the cell membrane also. Okay, now next, okay, after this, immerse it in a beaker containing alcohol. That means carefully place the above beaker in a water bath till the alcohol begins to boil. Now why this water bath is kept means a beaker containing water, and inside it then a test tube which contain ethanol, and then immerse the leaf in it. Why this is kept inside a water bath? Because alcohol may catch fire very easily if it is just boiled directly over the flame. And this boiling, why it is done? To decolorize the leaf, that is to remove the chlorophyll. Okay, so the green leaf becomes colorless by dipping it in the ethanol. So after this there is one question: what happens to the color of the leaf? So the green leaf becomes colorless. Why? Because there is no chlorophyll present. Chlorophyll has been removed. Now what is the color of the solution? That means this solution. So the color of this solution is green. Why? Because chlorophyll responsible for the green color gets dissolved in the alcohol, and thus the color of the alcohol turns green. Okay, now after this it is washed again, means it is put in hot water. Why? Because since the leaf becomes brittle after boiling in alcohol, so dipping it into hot water enables it to be softened. Then after this, pour the dilute iodine solution over the leaf. After 4 to 5 minutes, whatever the excess of the iodine is there, rinse it off and observe it. Now what is the observation? You what Now observe the color of the leaf and compare it with the tracing paper of the leaf done in the beginning. Okay, so what is the observation? The leaf has two type of patches. Okay, so bluish black and yellowish one. The bluish black area are the one which have starch, and the bluish black color is due to the reaction of iodine with starch. Now the yellow area are without starch. So when comparing the bluish black one and the pale areas, this one, and the green and the non green areas is sketched on the paper. So bluish black area are the one which are green previously, while non green areas remain pale colored. Now next question, what you concluded from it, okay, about the presence of starch? So the conclusion is that only chlorophyll containing areas produces starch, which is product of photosynthesis. Okay, and then this starch gives the iodine test, and thus these are the areas affected by the starch test which turns blue black. Okay, So therefore we can say chlorophyll is necessary for photosynthesis. Now next is given on page number 97, where it is a experiment set up with potassium hydroxide, because potassium hydroxide absorbs carbon dioxide. Okay, now this shows that carbon dioxide is necessary for photosynthesis. Now here two healthy potted plants are taken, then again they are kept in the dark room. Okay, now after this, each plant on the separate glass plate place a watch glass containing potassium hydroxide. Okay, one with the potassium hydroxide, other with not. Now the edges of the bell jar are sealed properly so that the carbon dioxide does not diffuse into them from outside. Okay, after this, keep the plant in sunlight for about 2 hours, and after this plug the leaf from both the plants and check for the presence of the starch. That means again you pour the iodine, that means dip the leaf in a dilute solution of iodine for a few minutes. Okay, now do the leaf show the presence of the same amount of starch? What you concluded? So first of all, the observation was that the leaf of set B, which was without potassium hydroxide solution, turns blue black, while the leaf of the other set A with potassium hydroxide remains pale colored. Okay, so both the leaves do not show the presence of same amount. Why? Because in photosynthesis, starch is produced using sunlight, chlorophyll and carbon dioxide, and in the first setup, availability of carbon dioxide will be less for making starch by the plant leaves, as potassium hydroxide absorbs the carbon dioxide. But in the second setup, the leaf will have more amount of starch because there is no potassium hydroxide, so no absorption of the carbon dioxide. Okay, now next one is, what can you conclude from this activity? Okay, so you can conclude that carbon dioxide is necessary for photosynthesis. Okay, so what is the conclusion arrived after observing the leaves? So the leaf of set B, which turns blue black, was synthesized starch with the help of photosynthesis. Okay, that means as there was carbon dioxide in bell jar. Okay, because there was no potassium hydroxide. Now this leaf of set A has not synthesized starch because photosynthesis does not occur, and the bell jar of this set does not contain carbon dioxide, as the same is absorbed by potassium hydroxide solutions. Okay, so we can say that carbon dioxide is necessary for photosynthesis, and it essentially affects the process and outcome of the photosynthesis. Now next is the test of saliva on starch, that is on page number 99. Okay, where it says, take 1 ml of starch solution into 2 test tube A and B and 1 ml saliva to test tube A and leave both test tube undisturbed for 20 to 30 minutes. So 1 test tube A and test tube B, in both starch solution are taken, but in only 1 ml of saliva is added in test tube A and leave this both for 20 to 30 minutes. Now add a few drops of dilute solution to the test tube. Okay, so in which test tube do you observe a color change? So you will see a color change in the test tube B, which shows the presence of starch because of the iodine test, and whereas the test tube A shows no color because due to the addition of saliva, because starch is converted into sugar. Why? Because the salivary amylase which is present in saliva has broken down the starch into soluble sugar. Now what does this indicate for the presence or absence of starch? It indicates that test tube A does not contain starch and test tube B does contain starch. Why test tube A does not contain starch? Because it is used up, that means the saliva has broken down this starch into soluble sugar. Okay, so what does this experiment tell us about the action of saliva on starch? This says that on addition of saliva in starch solution, the starch gets converted into the soluble sugar. So this all the 3 activities have been finished. Now these are the question. Do it in your note copy. Everything is clearly explained to you. In the next class we will start with the respiration topic. Read NCERT till here. Okay, that's all for today. Thank you.